This Week in Virology, the podcast about viruses, the kind that make you sick.
Bom, em primeiro lugar, eu queria agradecer a presença de todos e explicar, talvez, para algumas pessoas que talvez não saibam o que é o This Week in Virology, que isso é um podcast que é produção, execução, é one-man show do Vincent e hoje está sendo ajudado pelo professor Grant McFadden. E trata-se de um podcast, portanto vai ser colocado na internet. Aliás, ele está sendo ao vivo na internet ou não?
Is it online?
Is it online?
No.
Like people can see it in real time?
Not now.
Not now, okay. Ok, então ele vai ser colocado na internet logo depois e todo mundo do mundo inteiro pode acessar esse podcast, tá? E é uma coisa informal, trata-se mais ou menos de uma conversa, de uma entrevista sobre vírus, sobre o que a gente faz, e então a gente está honrado de ter sido convidado para isso, ok? So I want to thank you, Vincent, and thank you, Grant, for this. Então, essa é... Essa semana na Virologia, um podcast sobre vírus, mas do tipo de vírus que lhe deixa doente, tá?
Thank you. It's time for This Week in Virology, episode number 155, recorded October 24th, 2011. Hi everybody, I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Today we're recording live at the 22nd National Virology Society meeting in Atibaia, São Paulo, Brazil, before an audience of virologists. And joining me today from the University of Florida at Gainesville, Grant McFadden.
Obrigado, Vince. And thank you for not saying I'm Rich Condit, because I'm not.
No, he's not Rich Condit, but he is a pox virologist. He is from the University of Florida at Gainesville, and he loves talking about viruses. So that's why he's here today. And we have some special guests today, virologists who work here in Brazil. And starting on Grant's left, Paulo Eduardo Brandão, professor in the College of Veterinary Medicine at the University of São Paulo, Brazil. Welcome to TWiV.
Thank you.
And thanks for joining us.
Thank you for the invitation.
Did I pronounce your name?
Sorry?
I pronounced your name correctly?
Almost.
Almost. How should I say it?
Paulo Eduardo Brandão.
Good. I'll splice that. I'll splice the audio in when I say it. To his left, a professor at the Universidade Federal do Estado de Rio de Janeiro.
No.
Sorry. No?
No.
That's what's written on the piece of paper that they gave me. Okay, try again. University of São Paulo?
University of São Paulo em Ribeirão Preto.
You know, on the paper they gave me, it's the other place.
I'm sorry.
Maybe you're changing?
But I am not from there, sorry.
I'm sorry. Okay, so anyway, his name is Eurico Arruda, and he's a professor at the University of São Paulo. Welcome to TWiV.
Thank you for having me, Vincent.
Eurico is the reason why I'm here. He invited me to come to this meeting. He invited me to give a keynote lecture, and he said, why don't you do a TWiV while you're here? So here we are.
Good.
To his left, see if I get this one right, Francisco Murilo Zerbini in the Universidade Federal de Viçosa.
Viçosa.
What?
Viçosa.
Viçosa?
Yes.
Right. How do you spell that?
Vicosa, but the C has a little...
The C has a
something.
little thing in it. Yeah. It's missing on my computer here. Anyway, Francisco, welcome to TWiV.
Thanks a lot for having me.
And I'm very sorry I mispronounced the name.
I'm very used to it.
But that's the correct university, right?
Yes, that's right.
All right. I'm very nervous now. I get them all wrong.
Just wait to get to mine.
Last but not least, Janice Zanella from Embrapa Swine and Poultry, Brasilia.
Yes.
Is that correct?
Actually, Concordia is the place. But, you know, most of it all is correct.
So
Thank you.
Concordia is...
It's Santa Catarina State, south of Brazil.
Okay. So no more Embrapa?
Yes, in Brazil.
All right. Anyway,
It's
welcome
Embrapa.
to
It's
TWiV.
Embrapa.
Just leave it.
It is Embrapa?
It's Embrapa. It's
Okay.
a different state.
Yes.
Thank you for coming.
Thank you for the invitation.
So, of course, what we're going to do today is talk about what these individuals do. And we have a wonderful audience here as well, I want to point out. And you can ask questions if you'd like. There are individuals with microphones here. So if you would like to ask either now or during the show or at the end, you're welcome to do that.
And Vince, you should point out there's a lot of them.
A lot of microphones?
No, a lot of audience.
There is. The room is almost full. I would say there are 500 people here. How many of you have heard of TWiV before? Could you raise your hand? Half the audience?
More than half.
Okay, I'll take it. Very good. Well, this is what we're doing. We're doing a live TWiV today. This will be recorded. It's being recorded, video and audio. It will be put on the internet, and probably over 10,000 people will watch or listen to this at some point in time. So we're educating the public about viruses by doing this. So I first want to start, Grant, we've had you on TWiV before. We know where you're from and how you got there. So let's have each of our guests tell us what was their training that brought them to this point. We'll start with you, Paulo.
Okay, thanks for my training, right?
Yep.
So I started as a undergrad student, and I got my degree on veterinary medicine in 1997, and then I took my master's and my PhD on epidemiology, and then I worked mainly with rotaviruses and coronaviruses. So that's when my interest on virology started.
And all of the training was here in Brazil?
Up to the PhD, yes. Then I spent like 2 months at the CDC.
CDC?
Yes.
In
After
Atlanta?
Sorry?
Atlanta?
Atlanta, yeah, that's it.
And then the position you're in now was your first position?
No, it's my second position. I was for 2 years and 2 months a research scientist at Pasteur Institute in Brazil. So then, from then on, I also work on rabies.
Okay. All right, great. How about you, Eurico?
Well, I am an MD who gave up patients to go to the laboratory. So I did training, clinical training and clinical work, but then I decided to work with viruses. They are much more interesting than people. And, so I had a PhD here in Brazil, the Federal University of São Paulo, and that's probably why you had a federal name before my name. And then I went to the US for a postdoc. And then I came back and...
Where in the US?
In Virginia, Charlottesville.
Oh, Charlottesville.
At the Common Cold unit there with Fred Hayden and Jack Waltney.
Why did you decide to give up medicine? Because viruses are more interesting?
Oh, definitely. They are certainly much more interesting. And viruses are wonderful, and the intricacy of that biochemical parasitism was fascinating to me. So, no, I don't want to do anything.
So you have no further contact with patients?
No. I see samples.
Yes.
Does that count?
Yeah, that's fine. You don't have to actually treat patients, and you don't miss it either.
Oh.
Okay, very good. Francisco, what's your training?
I'm actually an agronomist.
Agronomist.
Probably very different from other people on the table. I got my degree in Viçosa in 1988, and when I decided to study agronomy, I really didn't know what it was, and for the first couple of years, I wasn't sure why I was doing it.
What is agronomy?
I still don't know. I'm kidding. Well, I'm just saying. It's a very broad course. You see everything from animals, animal production, crop production, and I learned very quickly that I didn't want to work with animal production, and started to move into plants. And then one day I was watching a lecture on plant viruses. I just had this epiphany, and I said, I want to work with these things. They're fascinating. And then I got a master's degree on plant pathology, already working with viruses, plant viruses. And then I went to the US for my PhD at California, University of California, Davis. Got my PhD there, also with plant viruses. And came back to Brazil, and I've been in Viçosa since 1996. We don't move around very much here, as opposed to the US, so I've been in Viçosa ever since.
How did you like California?
California was wonderful.
I'm surprised you didn't
Back in 1996.
I'm surprised you didn't stay. Many people are seduced by California.
You know, I was after 5 years, I was ready to come back.
Okay, very good. Janice, about you?
Yes, I got my veterinarian degree in Minas Gerais state. People know me, think that I might be from Rio Grande do Sul, or being a gaucho because of my accent. But I'm from Minas Gerais, I'm from Varginha. People here, yes. So maybe this is why I'm here today. You know, so lucky.
That's where the extraterrestrial
Because
people
there's
come
a extraterrestrian, adventure some years ago there. So my town became very famous because of that. So. But I decided to go to vet school. First of all, because I was raised in a farm. And I just loved the animals, and I thought that was going to be just that. But then when I was studying for vestibular, that is a big test before you get to vet school, or to college here in Brazil, especially to public college. I was studying pathogenesis. Actually, I was studying parasitology, the cycle of toxoplasma and all those parasites. And I thought, my God, this is divine. This is just great. And I have to do something that's related with pathogenesis. But that time, of course, I didn't know. I was not, I didn't have the experience maybe, but I thought that was something that I was going to do forever. And today, so I got my master's, I got my vet degree. I went as a trainee to Embrapa. I spent there a couple of years. I got some experience working for an industry, agribusiness, also in Brazil. And I decided to go do research. I was not, industry was not my field. And I got my master's and my PhD in Nebraska.
Right.
My PhD professor is Clinton Jones. He's going to be here on Wednesday to give a talk in herpes virus latency. And after that, I came back to Brazil, started working in Embrapa as a researcher. So my field is swine virology. In a couple of years, I had the opportunity to go as a sabbatical to US, but now to a colder place, to Iowa, Ames, Iowa. So I got to go there for 2 years, and I was in the right place at the right time, and I got the opportunity to work with influenza.
Right.
And when the pandemic
Sure. Hard to avoid pigs.
emerged.
Yeah.
So
in Nebraska, I can understand why you didn't want to stay there.
actually, I like it.
You liked it? I know. I shouldn't say that because probably we have a lot of listeners in Nebraska. My apologies.
I'm sure you'll hear from some of them events.
Yes, Nebraska is wonderful, wonderful state. You should visit. Absolutely.
In the winter.
But you said Iowa is colder than Nebraska?
Oh, yes.
Really?
More ice.
It's another wonderful state, Iowa. You should try it.
It's just Wisconsin now. My next one is going to be Wisconsin.
Yeah.
Maybe it's going to be colder.
So what's interesting is that many people have a moment in their life where they say, I love this, I have to do it. We hear that often. That's why I like to find out what motivated people to do what they do. And they say, oh, I just thought this was so interesting. And that's the best reason to do something, that you think it's interesting. So thank you all for that. Let's start with Polo and talk about your work. I wanted to talk about your work on infectious bronchitis virus.
Not sure.
Would that be good for you
Sure,
to do?
absolutely. That'd be
I
good.
hope so, because that's all I know about.
I'll try to prove it's a quite
So
interesting field.
Tell us the basic virology. What is IBV?
Okay, it's a coronavirus. A coronavirus is a very large virus, and its name comes from a crown, right?
Right.
So you can call it a crowned virus.
Right.
And it's a virus that transmits quite fastly among a flock of birds. So it's a quite important, from one side, it's a quite important disease for the poultry industry. But by the other side, it's also an amazing virus to study basic virology due to its complex replication system, to its complex transmission, and to the intricate host relationships you can depict, right?
So I understand that the poultry industry is important here in Brazil.
Yes, it is. Like, for instance, we have like per year 100 million broilers, and Brazil is second on the world on poultry meat exportation. And if you go to a layers farm, you can have like millions and millions of birds all crowded together. So this makes this virus amazingly diverse. I can say that every day it might have a different viral population there due to its high rate of transmission and high host density as well.
So what does it do to a chicken when you have IBV in a flock?
You don't do anything with that sick chicken. You do with the other ones. You try to vaccinate prior to the disease gets to the flock, right? So it's mainly prevention, not cure.
So
right?
obviously the sick chickens you can't sell.
No, you can sell.
Do you kill them?
You gotta kill them, or they might be either discarded, or they might be.
Right. What is the pathogenesis of the virus in the chicken? What does it do to the animal?
Okay, there are like many different lineages, right, of the virus. So all of them begin replicating in the trachea. So depending on the lineage, the sublineage, and on the parotype, the virus might reach the kidneys or the reproductive tract or the gut or lungs, and depending on each site, you might have respiratory disease, you might have diarrhea, or you might have nephritis, you might have like decreased egg production due to affection of the reproductive system as well.
And when a flock acquires the virus, where does it typically come from?
From other flocks.
Other flocks.
Unprotected flocks. When you think about large populations, the population is the individual, right? So the reservoir is another flock, an unprotected one. So
it is not a zoonotic infection?
No, it's not. We have some coronavirus that are zoonotic, but not this one.
Actually,
absolutely not.
It does infect animals, but it doesn't come from other animals other than chickens. That's what I meant to say.
Oh, so no, there are other chickens. Like you might have the very same virus in pigeons, quails, turkeys, and ducks and geese.
So is there evidence that it would spread from a wild bird to domestic chickens?
There's some evidence when you have the introduction of a new lineage.
Yeah.
Like if a new lineage appears here from Brazil and comes like completely out of the blue, you don't know where it came from. This has been suggested as related to migratory birds that might bring those new lineages here, right?
Right. So is this present all over the world, wherever chickens
yes. Every flock, every country has IBV, and every country has its own lineage as well.
So is there a vaccine that you can use to prevent spread?
Yes, there is a vaccine. Many countries use more adequate vaccines. Many countries don't do that. But there are a lot of different vaccines, different vaccination schedules you can use to protect the birds.
So is it given to all birds, or do you wait for an outbreak and then immunize?
No, you gotta give to every bird. If you don't do that, and if the vaccine is not killed, it's attenuated, the vaccine virus might cause disease. So we gotta vaccinate all of them at once.
I see. So this is an attenuated vaccine,
yes,
which is shed by the birds.
That's it.
And if you have unimmunized birds, they will be infected.
They'll be sick by vaccine.
Wow.
That's true.
So then when you immunize, so the outbreaks, do they occur in flocks that are not immunized? Is that the problem?
You don't see like outbreaks because the disease is endemic, right? You see that like it's present worldwide and in a endemic level.
So what does your work do that would impact the virus in chickens?
how my work would impact.
Yes.
Okay, mainly from one side is to try to do some molecular epidemiology, like, okay, how many lineages do we have here? How close are they to the vaccine lineage?
Right.
Can we infer any protection from this sequence data? And on the other side, we're trying to assess how proteins not involved in protection might guide the disease to one side or other, like how viruses affect the infected cell, and how does this become disease, right?
So do you think that at some point in the future you might develop an alternative to the vaccine that could be used to interrupt transmission?
Oh, sure. There's some research going on how you could use RNA interference to control it, not in very large populations, but like in high-value birds as the, how do you say, grandparents of the birds with high genetic value.
I see.
They are kept in smaller flocks, so you can use the most costly measures to these ones, right?
So how would you deliver RNAi to those birds?
Okay, this is a good question. You can a very effective way to do that should be using a viral vector. So we have started this in vitro, right? But not using viral vectors yet, only naked RNA.
Yeah.
But this should be one idea, or a more complex idea should be to do a transgenic chicken that could express that RNA as a endogenous way, and you would have a resistant lineage of birds, right? Should be an idea.
So in the U.S., if you propose to infect chickens with a virus vector or make them transgenic, you would have a lot of problems.
I'm sure.
Consumers. Is it the same here?
I know. Yes, it is. Yes, it is. Of course.
Is it worse?
Yeah.
Worse.
So that would be a problem, right?
That would be, but maybe someday scientists would rule the world. So.
I don't think we would, but maybe we could spread the word and educate people so that they understand the value of such approaches.
Paulo, I was looking at some of your papers, and I noticed you studied some viruses in bats.
In bats, yes.
Can you tell us a little bit about it?
Sure. The idea is that the SARS outbreak, right, that came up on 2002, November, something in Southeastern Asia, that coronavirus came from bats, right?
Correct.
So bats are the real reservoirs of real ancestors of every coronavirus known so far. So what we did is we checked for coronaviruses in vampire bats,
right?
And we found a virus quite similar to some human viruses.
Was it similar to SARS, or was it very different?
Nothing to do with SARS.
Right.
So you think it's a precursor to the?
I don't think it's we didn't find the precursor, right? It's like a more modern virus, I guess, but it has nothing to do with SARS as far as we know, right?
Right, because bats are suspected as being, mixing vessels for many viruses, and it's amazing the diversity of viruses in bats.
And also the diversity of bats itself is amazing, right? You have like maybe 1,200 different species in the world.
Yeah, absolutely. In fact, I think a bat was blamed for the virus in the movie Contagion. I don't know if any of you have seen it.
Yep, not yet.
Oh, it's not here in Brazil yet. Well, you have to go see it. Well, maybe I blew the plot line. Sorry.
I haven't seen it yet. Have you? You've seen
Yes. Actually, Rich Condit and I and a few other virologists went to see it together, Nissan Masach, and we all did a showing of the movie.
Yeah, we'll discuss it one day on TWiV, but we haven't seen it yet. So I haven't seen it, so we can't do that. So what are you going to be doing in 10 years?
Good question.
Well, what I'm going to be doing in 10 years, I guess I'll be arguing with some legal issues about this idea of transfecting birds.
Okay.
So the lawyers will get rich.
I guess that should be
Could you
possibility.
think it's too bad you couldn't think of a more pleasant thing to be doing.
No,
What
no,
about
I'm kidding. No.
In science, though, what would you be doing?
I'm sure I'll be going deeper and deeper on coronavirus, of course.
So what would you say the fraction of your lab works towards practical applications of research versus basic research?
Sorry?
So your lab works a little bit on basic
sure.
virology and a little bit on...
We're starting to go more basic now.
Yeah.
We used it to be like more applied because it was like easier to do, right? But we're going basic now.
Yeah. Is there pressure in Brazil to do translational
Yes.
There is.
Oh
research?
yes, yeah, absolutely yes. There is. You're right. It's hard to be basic.
Yeah, it's a problem, as I said the other day as well, in the U.S. So you have to keep, the only way to fight that is to have interesting basic results.
Sure. Let's try.
Anything else for Paulo before we go on? Eurico.
Ooh,
I'm sorry I got your university wrong.
that's no problem.
Now, I would like to talk a little bit about bocaviruses with you.
Boca.
Is that okay with you?
Yes, please.
Why are they called bocaviruses?
Because they fall in this cluster of parvoviruses that include the bovine and canine. So boca. It's nothing to do with the mouth, which is boca in Portuguese.
Yeah, okay. Bovine and canine.
Yeah.
Okay. And where were they first discovered, under what circumstances?
Oh yeah, this guy in Sweden, Tobias Allender,
Yeah.
he did this metagenomic study with samples that had been tested negative for all respiratory viruses, but they were kept frozen by someone very careful. And that's something we need to keep in mind, that we need this in labs. So he went back there and made a pool, and through this metagenomic analysis, he discovered this new parvovirus, which he named human bocavirus, as opposed to the bovine and canine and the other porcine and other bocaviruses in that cluster.
So this is a parvovirus.
It's a parvovirus.
Small single-stranded DNA. Right?
Yeah, single-stranded DNA, very, very small, naked, and several important parvoviruses had been studied a lot before that, like the B19,
Right.
like the dependoviruses, adeno-associated viruses.
Sure.
And our interest in bocavirus was more like, what is this virus really doing? Is it a pathogen? Does it cause disease? Or even, is it any important? Or are we just finding this passerby?
Is it found globally now since that first discovery?
Yes, everywhere,
Yeah.
every continent, almost every country has tested for that. It goes from anywhere from 1 to 2% up to 20% in kids with respiratory disease or with diarrhea.
As measured by seroconversion?
As
or
measured by PCR, by detecting
PCR.
genome.
So it's
yeah.
genome.
Yeah, that's one issue, a very important issue.
So it's active genome, in other words?
Yeah, well, no. And this is the point of our interest right now, is people started finding bocavirus frequently, and frequently associated with other viruses, other respiratory viruses. So was it the pathogen, or was just being shed by the kid while the disease was being caused by other virus? So we studied something like 1,200 samples or 1,000 samples in the hospital at the university where we work. And we retrospectively analyzed those samples and found 50 of them positive for bocavirus. And to make a long story short, we decided to test for messenger RNA for the virus in the secretion as a marker of active replication rather than just merely detecting the genome. Mm-hmm. And we found, to me it was very surprising, that only 1/4 of the kids were shedding messenger RNA for the capsid protein of the virus. And of course there is the issue of stability of the RNA in the secretion, so maybe we are not as sensitive as we should be because of that. But I can tell you that less than we find a lot of Bocas, but in my opinion, less than half, maybe 1/4 of those are actually infected, acutely infected.
Right.
And the others should be shedding some kind of form of DNA, like an episome, or maybe shedding cells that contain the episome in the nucleus. And as PCR, real-time PCR is so sensitive that you would pick that up.
So you think it could be latent for some period of time?
Yeah, there is latency in parvoviruses, but there is no mechanism studied and proven for Bocas. But the lesson we learned from the family is that latency is certainly a possibility, but also persistence. And more recently, since the virus has this reputation for being persistent or latent, we went for chronic disease. And then those kids that have hypertrophic adenoids and palatine tonsils, removed by the otolaryngologists, the ENT doctors, they send us the tissue to the lab and we search for viruses. And we test for all the known respiratory viruses and by real-time PCR. We plan to do deep sequencing someday, but right now we test for this panel of respiratory viruses. And to me, it's very startling that we found 97% of like 120 adenoids that we tested, 97% of them had at least one virus, frequently 2, 3, 4. And those kids do not have acute disease when they go to surgery. So I think we are in for to me, a very important lesson about persistence of viruses in lymphoid tissue of the upper respiratory tract.
So this is, one possibility is that this is just a carried virus. Most of the time it's of no consequence.
Well, it's a carrier state maybe of some of them, but I wouldn't say without consequence, because I think we are just starting to understand that like episomes and other forms of RNA viruses that can stay and persist in a cell can stimulate chronically the innate immunity or the innate response of the cell. And how does that relate to chronic inflammation and hypertrophy in the long run? So those kids go to surgery very often to remove adenoids and tonsils. So why is that? Could that be that viruses stay in those lymphs and stimulate the hypertrophy, production of collagen and fibroblasts and whatever?
So it sounds to me, though, that you don't think it has a role in respiratory disease per se.
I think it has a role that's less than propagated. Okay. I think it's important, but probably important for things that we are yet to discover.
And so what's the significance of virus recovered from fecal material associated with gastroenteritis?
Yeah, that's another question. This virus is the relationship, the causality of disease by bocaviruses has not been proven, not even respiratory or digestive. So maybe the same kind of thinking can apply to the gut. And lots of kids shed bocaviruses. And only now we are learning that there are at least, I think it's 5 different species, different genotypes. I think they are calling it species. The one is the more frequently detected in the respiratory tract, and the others are more frequently related to the digestive tract and detected in stools.
So is it possible that in the GI tract, the virus is in lymphoid tissue as well as it is in the...
Yeah. How about that?
Yeah.
We have to study that.
You have to go in and snip out some lymphoid tissue.
That will be difficult, but...
Why?
Yeah. Well, there is no animal model, and how do you go about getting patches from the gut of someone? It's hard. Intestinal biopsies or something?
And nobody's donating.
Not really.
Well, you can take intestinal biopsies for various purposes, but
yes, for
maybe
sure.
the problem is to find a virus patch, right?
Yeah, and see which one is hypertrophic and We could try that, certainly. It's a good idea. Thank you, Vincent.
Well, you're a physician. You're not a gastroenterologist.
No.
You'd have to
I'm
collaborate
not a
with
surgeon.
one. Yeah. So, but that might be worth looking, because it sounds to me, and this aspect that it could be in lymphoid tissues is new. It could be that it's there chronically, and then when you have a respiratory infection, you isolate it. It's been there all along.
Yeah, but you know, parvoviruses being latent or persistent in hematopoietic tissue is not news. So if bocavirus does the same thing, right there you have a mechanism.
Sure.
Is there any evidence that when patients are under immunosuppression, that bocavirus starts to grow at higher levels?
Not that I know of. I don't think so. I mean, that's another good idea, to study the
Like transplant patients?
Patients, AIDS patients, or...
Right.
Yep. So it sounds like we're a long way away from a vaccine.
Oh, certainly. Certainly. And maybe I don't know that you would sell a vaccine for bocaviruses too easily, because they don't seem to be very pathogenic per se, like don't cause very serious disease. But if we prove that bocaviruses are associated at least with some portion of hypertrophy of adenoids and tonsils, that would be good.
Yeah. It's a good lesson on how simply isolating viruses from patients with certain disease doesn't prove causality at all. It's just an association, and you need to do a lot more. Are there a lot of individuals working on this problem globally?
For bocavirus?
Yeah.
Not that I know of. Few people only working with bocaviruses. There is no model, at least that I know of, animal model. And it doesn't grow in cell cultures. Someone grew it in this very special kind of primary cell that blah, blah, blah. But there is no regular routine cell line that will support it. But there is reverse genetics already done that could help.
Grant, anything from you?
Well, one thing that sort of fascinates me about it is that it's single-strand DNA circle. Is that right? In the genome? Well, I did my PhD on XXII, a bacteriophage single-strand DNA circular virus. And I've always been fascinated why Mother Nature chooses that as a genome in a virion. Any thoughts?
Well, excellent question, but I don't know the answer.
I don't either.
But some things fascinate me about the structure of this episome, because they form these concatenames inside the nucleus before they get sliced up to be the genomes of the progeny. And some of the concatenames are either like this or like this, so they come in both flavors. And actually the virus can encapsulate both the minus or the plus DNA, so everything goes.
And the concatenamers are single-strand DNA or the double-strand?
No, they are
replicative
circular.
Form?
Yeah. They can become circular in, I think it's in 2 or in 4 or in different numbers of copies.
But as single-stranded DNA or double-stranded when they're actually concatemerized?
They are, how do you call that? It's a double-stranded, but it's closed at the end, so it's a circle.
Yeah. So it's the replicative form of the virus. Yeah.
So my answer, Grant, would be that it works. That's why it's done.
Absolutely. Mother Nature has tested all kinds of things, and she finds it works.
Has tested double-stranded RNA.
Yeah. Who are we to argue?
Yeah.
Well, so we know of 7 different viral genome types now, right? There's 7 different kinds of nucleic acid. That's according to the Baltimore scheme.
Yeah.
And a good question is, are there any that haven't been found that could exist other than those 7?
Well,
you don't have to answer it.
That's a good exam question.
It's a good exam question. It's a good thought question. It might even be a Talmudic question. I don't know.
Talmudic.
I don't know. Anyway, let's move on to Francisco. I like your last name, Zerbini. This is of Italian origin?
Yes, it is, yeah.
So you're way back in history, someone came from Italy in your family.
Yeah, my great-grandfather. I'm 25% Italian.
Very good. It shows.
Yeah.
I wanted to talk to you about I'm going to say this wrong. Begomoviruses?
Yes.
Begomo.
Single-stranded DNA viruses as well.
They're single-stranded DNA. They're circles, right?
Yes, yes.
But they're in 2 pieces?
For begomoviruses, there are monopartite and they're bipartite.
Oh, they're both?
There's
Okay.
both types, yeah.
So I was really interested in the one that is transmitted by whiteflies.
Every begomovirus is whitefly transmitted.
Every one of them
Yes,
is?
yes.
And those are, how many circles do they have, the
Well,
whitefly virus?
Okay, let's go. You're talking about begomoviruses. That's a genus in the family Geminiviridae.
Yeah, and this one,
Okay.
Cleome leaf crumple virus.
Cleome leaf crumple.
That's a begomovirus.
That's a begomovirus, yes. That's one that we've recently described in my lab, yeah. Geminiviruses, the family. You have leaf hopper transmitted viruses and you have whitefly transmitted viruses.
Okay.
The whitefly transmitted viruses are called begomoviruses.
And the leaf hopper?
The leaf hoppers, there's 3 different genera.
Okay.
Mastrovirus, curtovirus, and tuberculovirus.
Okay.
They're all leaf hopper transmitted. So let's forget about them.
Let's
forget
do the
about it.
whiteflies.
Let's talk about soccer.
Let's forget about the leaf hopper transmitted viruses. The whitefly transmitted viruses are begomoviruses, and there's monopartite and bipartite.
Okay.
In the Americas, you only have bipartite viruses. There's one exception, but there's bipartite viruses. The monopartite are all in the old world: Africa, Asia, Australasia. In the Americas, you have the bipartites. And the one you just mentioned is a typical new world, bipartite, whitefly transmitted begomovirus.
So you see, we don't often talk about plant viruses on TWiV. So we have to do it more often, don't you think?
Of course.
I think you're only the second plant virologist we've actually had.
Who was the first?
Marilyn Rusink.
Okay.
Ooh.
Okay.
And so we have to do it more often. You can see I don't know much. I like whiteflies because we just did a story on one of my other podcasts about how in the southwestern U.S. and maybe also other places, they are being colonized by rickettsia.
Okay.
Do you know this story?
Not in detail.
So the rickettsia, of course, are intracellular bacteria. They're endosymbionts. And they move very quickly through the whitefly population because they confer female reproductive advantage.
Okay.
Sort of like Wolbachia.
The Wolbachia and mosquito thing, yeah.
And so in one study in the southwest U.S., California, Arizona, within 10 years, the rickettsia completely spread through the population. So I learned from reading about your work that these same whiteflies have these viruses in them. And I just wonder if there's any symbiosis between the virus and the rickettsia. That would be interesting.
About the rickettsia, I don't know. But begomoviruses, in order to be transmitted by the whiteflies, they need a protein that is secreted by a bacterial symbiont called Buchnera.
Buchnera.
From
I know that one, yeah.
Relative to E. coli.
Yeah.
So Buchnera secretes a protein, a chaperone. And this chaperone is essential for the virus to be, it binds the viral coat protein.
Okay.
And it protects the virus from degradation during its passage through to the insect intestine.
So where is the bacterium? In the insect's gut?
Yes, in the midgut, hindgut, I think.
Hindgut of the whitefly.
Hindgut, yeah.
That's interesting. I wonder what the presence of rickettsia would do, or if it's excluded by Buchneria or what. That's really interesting.
It would be a wonderful biocontrol tool, right?
Right. All right. So the virus, if I understand it, is present in weeds of different kinds, right?
Well, the begomovirus genus has a very large number of species.
Okay.
And most of them that, of course, we studied first, the one that infects crops. That's the ones that are important economically.
Right.
But we know now, we've known for a number of years that these viruses are very, very common in wild, we call them weeds, but they're actually wild, non-cultivated plants. Weeds is a very anthropocentric word. Right?
Yeah, it's perjurative.
Yeah.
Yeah, it is.
So, and these days we have tools and the means to actually investigate viruses in wild plants. So we're finding more and more of them.
So in the wild plants, I won't call them weeds. I'll try not to. In the wild plants, the virus does not affect the well-being of the plant?
No, they cause symptoms.
It does.
The growth reduction, they're pretty much the same thing that happens with the crops, except that it doesn't happen on a very large scale because you don't have 10,000 hectares of weeds being cultivated. You know, they're growing very sparsely. So you don't have that dramatic effect that you have when a virus infects a crop plant.
So I must admit I'm biased by Marilyn Rusing, who tried to convince me when she was on TWiV that all these plant viruses are really good for plants. And you know, the story of the
plant pathologist.
The heat tolerance in the grasses that are colonized by the fungus is an example, and she has many others. But I'm hearing from you that even in weeds, these viruses can be detrimental.
Well, as an agronomist, right, I tend to think about plants needing to grow as much as they can, being as productive as they can, and viruses interfere with that. So, a tomato plant infected with a virus will produce less tomatoes and smaller tomatoes than a healthy plant.
Right.
So, from the agricultural point of view, that's bad. But who knows in terms of evolution, right?
So what happens here is the virus is in weeds and it gets transmitted to crops by whiteflies,
by the bacteria.
in this case. Can you track this spread by sequencing the viruses?
We try to do that. And it's very interesting because we've been looking at the viruses in the weeds and in the crops, of course, and mostly you find different viruses in the weeds and in the crops. They're not the same. So something happens between the virus being in the weeds and going to the crop plant. It changes somehow. There's probably a very diverse population of viruses in the weeds. And you know, the quasi-species concept doesn't really apply to DNA viruses, but for the Gemini virus, it's starting to look that it actually applies. So you probably have a very diverse population in the weeds, and one of those minor variants is probably selected when it goes to the crop plant, and then it predominates in the crop plant.
The bottleneck, basically.
And you find a different virus in the crop plant.
Can you duplicate that in the lab? Can you put the virus in weeds and then have it go to tomatoes?
You would love to do that.
No, I can't do it. Why can't you do it? You've tried, I guess.
Well, a lot of people have tried, and it's an unsolved problem right now. A lot of people think that they can use deep sequencing to investigate that, but that hasn't materialized yet.
I'm sorry, I called them weeds. You didn't catch me.
Most of the time I call them weeds too.
So, I mean, you can grow these wild plants in the lab, right?
Actually, a colleague of mine just found that we can buy seeds from these weeds and grow healthy weeds in the lab. She's actually here.
Do healthy weeds become crops? I mean, when do we stop talking about weeds and calling crops? If you eat them.
The moment they become, of interest,
economical
food, or fiber crops.
So when you have a crop that's infested with this virus, what do you do?
After with plant viruses, you cannot do anything after it happens. You have to do it before. You have to prevent.
Okay.
There's nothing you can do afterwards except plowing the field and
try again.
Yeah.
So how do you prevent infection?
Vector control is one way.
Okay.
It works sometimes, but it's usually based on insecticides, so it's not very environmentally friendly. But it works. And a lot of it's very common.
Okay.
Another way is using virus-free propagative materials for certain crops where the viruses are transmitted by seeds or by tuber crops that you plant a potato to get a potato plant. And genetic resistance, the ideal way. Yeah.
Like transgenic plants that are resistant?
Well, conventionally, you can breed a resistant plant just by crossing, transgenics, and it's been done for many years. And now you also can use transgenics, yes, of course.
So why doesn't everyone use these bred crops to be resistant? Do the tomatoes not taste as good, or is there some
Well, I can give you an example of a begomovirus, a bean golden mosaic virus, very important here in this country. And people have looked at hundreds of, not hundreds, tens of thousands of wild Phaseolus accessions in germplasm banks, looking for sources of resistance, and they simply don't exist. So you cannot find Well, of course you can find resistant material, but then it's botanically unrelated to the common bean. You can cross them. There's a genetic barrier.
Yeah.
So in many cases you don't have natural resistance, and then therefore you cannot get a naturally resistant plant. Now, fortunately these days, and I tell this in my lectures, and just half jokingly, if by some reason tomorrow morning everybody wakes up and they think transgenics are fine, we could control every plant virus in maybe 6 months with transgenics, everyone. I would be out of a job.
It's amazing. You can control every
because the
plant viral disease, right?
Yes.
By transgenic
exactly.
technology.
The technology is there. You know, with RNA interference, you can clone a fragment of the viral genome as an inverted repeat,
right.
Transform a plant. As long as you can regenerate a plant in tissue culture, and you can do it to most plants these days, you can get an immune plant.
As long as you know what virus to immunize against.
Of course, yes, of course. Yeah. Every known viral disease, plant viral disease. Yeah. 6 months maybe is pushing it, but a couple years. Yeah.
So can I ask the audience, who would eat transgenic plants on a regular basis?
Whoa. See?
So not everybody, though.
Yep.
It's interesting. And
they don't know, but they're probably eating transgenics every day already.
Yeah, soybean.
Okay.
Is there a Brazilian word for frankenfood?
No.
Food for what?
Unfortunately, frankenfoods.
See? You didn't know what it was. That's good.
So you say if you devise an RNAi strategy, maybe you get another virus that comes in that's resistant to it. So that brings me to the question, do we know what the virome of the world, the plant virome of the world is? Yesterday I showed a slide showing in sewage, the biggest number of viruses comes from plants. Do we have a sense for how extensive that virome is?
We're finding new viruses all the time.
Yeah.
All the time.
Do you actually look in your lab for new viruses?
We do. Yeah, we've been describing new viruses all the time. Yeah. So I've in many other labs.
So if you go out and take a wild plant and bring it in and sequence it, you will find new viruses.
If I go to the field tomorrow, or if my students go to the field tomorrow, and they bring 100 samples, we can probably describe 2 or 3 new viruses in a couple months. We've been doing this for years. Yeah.
So the virome is huge, and it's likely that when you take away the known viruses by this strategy, more will come in. So it's a losing battle.
You have all these new technologies that are being developed that increase our power of detecting these unknown viruses. Yeah.
Sobering.
Yeah.
Anything else from you?
I'm sort of curious. I have to confess I didn't know very much about begomoviruses when I started reading some of your papers. But one thing I found intriguing is it's another small circular DNA virus. Yeah. Very interesting. But one thing I was curious about is you say that the virus can be either SAP transmissible or non-SAP transmissible.
That's true.
I found that really fascinating. Can you just sort of briefly describe that?
There is a pretty good correlation between a virus being phloem restricted in a plant and being SAP transmitted. So if many plant viruses, they can infect the phloem, the vessels that, like the blood vessels of a plant.
I sort of know.
Yeah. Most people don't. If a virus is phloem restricted, it will not be SAP transmitted. Other viruses, they can escape the phloem and infect other tissues in the plant, like the epidermis of the leaf and the parenchyma. These viruses usually will be SAP transmitted.
Huh.
Okay.
So, I mean, I'm Canadian and I grew up drinking maple syrup.
Maple syrup.
So after all these years, have I been sort of scarfing down begomoviruses?
If you eat plants, if you eat lettuce,
you eat viruses.
You're eating viruses.
Okay, fair enough.
Yeah.
Do you think plant viruses can replicate in people?
No.
If
they could, we would all be sick by now. Yeah.
There are some reports. There was one in a journal last year where they looked in peppers of various kinds, peppers that you would put on food, and they found a lot of viruses. And then people with GI problems, they isolated these pepper viruses from their feces, but you don't think
they're just there.
Yeah, just there.
Now there are many plant viruses that can actually replicate in their insect vectors. Okay, so they both plant and insect viruses.
But then what can you say about that, considering that this insect can suck a little virus from the plant, and inside the insect you have a bocavirus, which is tri-segmented, and then you can have recombination. And maybe you have just one segment of RNA there.
Sure.
You know, the plant virologists, they think that these insect-transmitted plant viruses, they were originally insect viruses that acquired the capacity to infect the plant. So it's actually the other way around.
Okay.
But who knows, right?
Right. Right. I've heard that one. That's a good one too. Okay, great. We've learned two really important things. One, you can cure all crop diseases with transgenic plants, and then the other is that plant viruses don't infect people. So that's the take-home message from your participation in TWiV today. Let's move on to Janice down there. Thank you for being patient.
I know, I'm learning a lot too.
Oh, good. That's very good. I wanted to talk about circoviruses with you, because we actually learned about them a year or two ago when they were found in some human vaccines. But these are pathogens of swine, right?
That's right.
So tell us a little bit about what they do.
Yes, actually, the beginning, the 70s, people knew about porcine circovirus. That was normal contaminant of tissue culture cells.
Right.
PK15, there are porcine kidney cells. So, and then there were contaminant cells. They were not pathogenic to pigs. These porcine circovirus, but 96, 97 in Canada, some researchers, actually there were some veterinarians, they went to a technical meeting, regional meeting, and they were demonstrating that, presenting a case actually about a disease, this new disease. And there was wasting disease, pigs in nurseries, they were just weaned and start to lose weight and get all these types of respiratory signs and die. Well, then took some researchers from Northern Ireland and also from Ohio State to find out that was a circovirus. And there was a pathogenic circovirus.
So that was the first association with pathogenicity?
With the disease. So they could reproduce the disease, and when they got sequencing analysis, they found out that was a pathogenic circovirus. And there was a big boom because many people didn't believe, because there's a say that if swine virology doesn't come out from Iowa State or Minnesota, nobody believes.
Nobody believes.
So it came from Europe and then Ohio State. So, and then was all these types of jokes in swine meetings, and also some magazines and some journals, technical magazines and journals, saying that was circus, like circus disease. But then after reproduction of the disease, the scientific community started to believe, and then and after now the vaccine, there are several commercial vaccines now. People. Well, it's accepted, but the Australians, they still don't, they say that they have the virus, but they don't have the disease. That's an extra agent, they call the XX agent, that is causing the disease. That is not the porcine circovirus type 2. So then the name was porcine circovirus type 2, because this was the pathogenic one. But then around, I think 2005, 2006, the Americans from Iowa and Minnesota, they start to believe, because they were. The problem is, it's not just that, because there are other pig diseases that are very important. They cause lots of losses, and they all curse together with porcine circovirus infections. So there are some reasons for this to occur, because when the amount of virus and some other contributors, so cofactors, help the virus to replicate faster. The animal gets the huge viral load and then gets sick. But for example, pigs from the same litter, one gets sick, the other one doesn't get sick. But they all get the virus, but in different rates. So anyhow, this is how all this disease became important, and today is the #1 porcine disease, I believe, in the world, because it was supposed to be an emergent virus, and is an emergent virus, but it's not a new virus. It's an emergent disease associated with this virus, and the disease has several flavors, like he said. You can see like this wasting type of the disease that is the most common, but there are other syndromes that can curse together, like dermatitis, nephropathy, and reproductive diseases. So
do we know where the virus originated, for example?
Well, that's nobody knows for sure. How it's like I was saying, in 2005, 2006 in US there was this different virus, what they call today PCV2A and PCV2B. Right? It's this one is the PCV2B. It is supposed to be more virulent, but nobody has ever shown that this is true, because you can have problems in farms that most of the virus is PCV2A, and sometimes you get problems with PCV2B. So in Brazil we try to associate with reproductive problems, sequence which virus it is. But there are some researchers from Denmark that found out another virus that they think that is the precursor. The first one is what they call PCV2C. In the 60s, there was some material that they had in the lab. They sequenced. There's some difference in nucleotides, but I mean, nobody saw that virus again. It was just in Denmark, and also researchers from China, they found out other viruses, PCV2D, PCV2E, but it's just in China.
But it's not been found in other animals besides pigs. Have people?
Well, actually, we have antibodies for PCV2. Humans, bovine, they have antibodies. Okay. But I mean, it is just pigs. Even sus scrofa and wild pigs or wild boars, they also carry this virus. But they don't have the disease as the same presentation as the commercial pigs, because they're more rustic.
So, did you say that pigs are routinely immunized for these viruses at the moment?
Well, yes. Now, yes. Brazil is #1 market for these vaccines.
And when you do immunize the swine,
they get better.
They're fine.
They're fine.
So do you immunize like for the chickens when they're born, or do you wait till the disease
comes?
Usually
there are several presentations of the vaccines. Yeah. Actually, there are 2 vaccines that are currently in the market. They are just the ORF2 of the virus, the capsid protein that is in baculovirus. So there's one that is 1 dose, there's another one that's 2 doses, depending. It usually is weaning. Pigs, we wean in 21 days, from 17, depending, because usually people get batches and wean. Yeah. So it's around 21 days. So usually they vaccinate before, but some farms, they associate the vaccine of PCV2 with mycoplasma. That is also causes respiratory problems in pigs. That's very common. So they vaccinate together just to save time and save labor. But there's another vaccine for sows and gilts. So this vaccine for sows helps to prevent reproductive problems in sows, and also it's a better colostrum quality. So the pigs, they get more prepared when they are weaned, and tends to decrease the circulation of the virus in the herd. But all the vaccines, they work great.
Sure. Anything from you?
I'm impressed by, this is another example of a small single-strand DNA circle. Isn't that impressive?
Yes. Yes. I was in a meeting in Barcelona in June, and there was this emerging disease, porcine emerging diseases, and there was, so there was a talk, it was a session actually, that was named something like, it's emerging, it's raining virus, so what? Right? So it's all this TWiV in pigs, this bocaviruses, even the hepatitis E virus, there are so many new viruses, the porcine epidemic diarrhea virus, there is all those, but most of those, for example, even TWiV or the bocavirus, nobody knows what the virus is doing there. Most of the pigs, they carry the virus. It's very hard to cultivate, it's no tissue culture, so, and many vaccines, and even, for example, trypsin that we use in the lab, they are contaminated with PCV2, TTV, all these, single-stranded DNA viruses that are, we don't know how this virus suddenly came and became important. It was in the past when this virus showed up first time. There was this argument, probably the virus came with a live vaccine, for example, a parvo vaccine, right? Maybe it was the way that the virus was distributed around the world, because it was around, because the virus showed up in Canada, in some countries in Europe, but in Brazil we got to diagnose the virus around the year 2000. But in 2003, it was like, spread all over. So nobody knows for sure if it was a vaccine or some byproduct or plasma, because there's some feeding additives. Dry plasma, like a spray-dried type of plasma, they are, if you do a PCR, you can detect it. PCV2. So.
Do you think it's of any harm to humans to have vaccines with circoviruses in them?
I'm not sure, but it's, at the same time, this virus is a DNA virus, right? It's very homologous. I don't think that is going to have a mutation and suddenly start to affect human cells. But at the same time, I don't like to think about it for us to get it, because this is a very nasty, it's a very nasty for young kids or young pigs. So you guys can imagine. So
well, it's very much like Boca, and could be it establishes persistent infections.
That's what I was thinking about that. It's a DNA circular, so it's double-stranded, but it's closed, so it's pretty resistant, right?
Yeah.
It's not like an RNA virus. You have to have a restriction enzyme to cut a double-stranded DNA, something that's lacking in eukaryotes.
That's right. And then we got to detect also this virus in porcine reproductive organs, like testicles, like some accessory glands in the bore, because the virus is transmitted by semen. So the virus is there, it's persistent, kind of a persistence in these tissues, but we got to see by in situ hybridization, the virus is there, but we don't know for sure what it's doing there. There's no escape mechanism, there's no apoptosis, we don't know.
So it sounds like most of us could have circoviruses in our cell cultures as well without knowing it, right?
That's right.
Because it's in the trypsin.
Yes.
So do we know the cell range of circos? Do they infect a lot of different cell types, or is it restricted?
Yes, in fact, several. Usually we grow the virus in porcine fibroblasts, but also infects some lymphocytes, infects macrophages, infects cells from the reproductive system. It is not just macrophages, like all the Sertoli cells, other cells. So we are not sure, I'm not sure if it is a true persistence, but the virus is there. The other problem is it doesn't cause any cytopathic effect.
Right.
So we have to do, do FAA to see if the virus is present.
Will it infect human cells in culture, like HeLa cells or?
Yes, HeLa cells, yes, infects HeLa cells.
We probably have it in...
Usually we use,
probably all have it
you know,
in your
usually
cultures.
in the lab you use D-glucosamine to stimulate the virus to grow faster, to get a better titer, but grows in HeLa cells, viral cells.
Someone should do the virome of HeLa cells, right?
And there you go. We may be in for some surprises.
Who knows what's in there?
Virome
The
of
thing
HeLa
is that, I mean, it sounds flippant, but if you have viruses there, they're not cytopathic, but they're replicating, or they're there, and they may influence your results that you get with other viruses. So it could be problematic, right?
Yep.
Any other questions there? We have a question from the audience. This is for Dr. Zerbini. Considering my presentation last night on on having virology alone, and of course all this other virology, how do you see the eradication of plant viruses with transgenics in this context?
In what context? I'm sorry.
Here, maybe you can read this, or give it to Eurico. If the person's still here, they could ask it in
He is right there.
I think I understand.
Okay, sorry.
Let me see if I actually understood.
Yeah, repeat the question.
Well, the question is, considering Vincent's presentation last night on marine virology alone, I mean, I guess you're talking about the
huge
magnitude
viroma.
of the size, the number of viruses, and
We need those viruses in
and
the
how do you
ocean.
see the eradication of plant viruses with transgenics in this context? Well, first of all, I'm talking about the eradication of plant viruses with transgenics in a hypothetical world where people will accept that. But anyway, and even if it happened, we're talking about eradicating viruses from crop plants, right? From agriculture. Viruses are still going to be infecting plants in the wild, right? In the forests, in the grasslands, you know, wherever plants are grown, they will be infected by viruses. I'm just talking about removing these pathogens from plants that we grow for food. That's what I'm talking about. Okay? Did I adequately answer your question?
I don't know. Perhaps if the transgenics would influence the evolution of these plants, that'd be a problem.
Well, maybe you're talking about gene flow, right? The transgene escaping from the transgenics and going to wild plants. That's always going to be an issue, and that's one of the reasons why transgenics are still not widely accepted by a lot of people, and it's a legitimate issue, absolutely. In my lab we've produced a transgenic passion fruit, which is immune to a passion fruit viral disease, and I am tentative about the possibility of planting this in a large scale, because what is it going to do to the wild passion flowers that we have in our country? So it's a legitimate issue, and we're going to spend many years debating this until we reach a conclusion.
Any other questions before we move on to our next part of TWiV? Okay. No questions from Brazil, huh? I got one? All right, great.
Hi.
Go ahead.
Now? Okay, work?
No.
Try it again.
Hello?
Okay.
It's work?
Yeah.
Okay.
Sorry,
Okay.
I'm a virologist.
A couple of TWiVs ago, you interviewed Rossink. I don't remember her first name. She works with plant viruses, and she told that there are some plant viruses that can help the plants to be more resistant to dry or to some harsh conditions. She made the work with the three-way symbiosis with a plant, a fungus, and a virus. So, is there some risk in eliminating plant viruses in this context?
It's for me again.
It's for you, yeah?
Well, I'm familiar with this work, and what I can say about this is that we're just starting to grasp the significance of the interaction between plants and viruses. Like I said, from an agronomist's point of view, we tend to look at viruses in a negative context. They're harming the plant, they're causing diseases, because we're thinking about the plant as a factory. We want the plant to produce fruit or things that we use. Now, from an evolutionary point of view, it's a completely different story, right? Maybe a plant that grows a smaller fruit will have a selective advantage over a plant that grows bigger fruit. Who knows, right? So when you find these systems where there's this symbiotic relationships and somehow the virus is helping the plant, that's a perfect example of that. But in an agricultural context, it's usually a negative effect. Now, of course, we have to look beyond the agricultural context when we consider these issues. And then, nature finds multiple ways of dealing with these different organisms. We have a very faint understanding of that.
I had enjoyed the idea of if you have some problem with your plant, instead of getting more water or something, you can apply a virus there and solve the problem.
It's not a reality yet, and I wonder how people would accept this idea of buying viruses to throw in their plants. We could probably make a lot of money from that. It's probably a few years from happening.
Any others? All right. One of the things we do on TWiV is we have a lot of interaction with our listeners. We get a lot of emails, so I would just like to read a few of them to you today to give you a sense of what they are like. The first one is from Kim, and she writes, "To the TWiverati intelligentsia." Very nice. We get different names each week, so that's a new one. "Each week you begin your show with the tagline, 'This Week in Virology, the podcast about viruses, the kind that make you sick.' I recognize that viruses have been responsible for some of the biggest epidemics and plagues of man, beast, and plant life. But as a longtime TWiV listener, it seems like you also cover viruses that have positive effects or impacts on our planet. Viruses that are used for varied things as batteries and to fight other pathogens or pathogenic responses. While the work being done on the microbiome suggests that keeping the correct balance of our symbiotic critters, including viruses, may be even more important than which ones are living inside us or on us. So even though it's catchy, maybe you should reconsider the tagline in an effort to cultivate respect for viruses instead of fear."
That's great.
"Thanks for the great 90 minutes of informative science and thoughtful discussion each week, no matter how you pitch it." And she says, "P.S. It's cloudy and 22 degrees Celsius." What's the weather here in where we are today? Do you know? Does anyone know the temperature?
Mid-20s and beautiful.
It's in the 20s.
Yeah.
It's very nice today. Clear skies. Well, this is interesting that I selected this email because we've been talking a lot about this today, beneficial viruses. To be honest, when I started TWiV 3 years ago, I wanted people to know this was about biological viruses, not computer viruses.
Right.
Because I knew there are a lot of podcasts out there that deal with those kinds of viruses, and I thought, how can I distinguish it? So I just had this idea, the kind that make you sick. And I know now very well that it's not always true, but I have to tell you, I love the tagline. I just love it. So I don't want to change it. It's just rhythmic and catchy, as she says. So,
and if I remember right, there was one TWiV where the tagline was "Viruses that make you better."
And that was the one you were on.
Yep, that's right.
Those were your viruses, exactly.
So you mean that no one ever pointed out that computer viruses can make you sick as well?
Is that right?
Anyone who's used Microsoft just pointed that out.
That's right. They can make you sick. They make you really sick. Exactly. The next one is from Luisa, who's here actually. She's a doctorate student from Federal University of Rio de Janeiro, and I'm going to attend the Brazilian Virology meeting next week. I'm also attending a discipline on my university about scientific divulgation, and the students should produce a podcast every week to post on the discipline website. I would like to know if you would accept to give me a short interview during the meeting next week. It would be an honor if we could have an interview with someone so concerned with scientific divulgation. In Brazil, people are still starting to give attention and importance to this matter. Hope you can accept it. So in fact, I did speak with Luisa today. We did a podcast, so you should look for it on the website.
Is Luisa here?
Is she here?
No.
Look at that, she didn't even come to TWiV. Luisa, I read this email so you would be flattered. Okay, well, we did a wonderful interview, and I think it's really important that students do this as well. And I told her, if you feel like doing a podcast or a blog, certainly do it. And I will read two more. Bear with me, because these are really cool. The first one is from Viniaka. The entire saga of the possible, now absent link between XMRV and CFS that has nearly concluded, as discussed in TWiV 150, reminded me of the need for scientists to have the right level of tenacity in their scientific belief. A fabulous guidance on how to do this is provided in an article by R.A. Littleton called The Nature of Knowledge, in a book I read very, very long ago, The Encyclopedia of Ignorance. Littleton graphically represents one's scientific belief in any concept or hypothesis as a bead on a straight line. The two extremes of this line are 0, complete disbelief, and 1, complete certainty. He argues that one should allow his or her bead to move in either direction based on the accumulation of evidence in favor or against. For example, if you believe greatly in favor of a concept, you allow your bead to approach 1. When evidence disproving that concept is presented, you should be able to move towards the opposite direction. However, he cautions that at the very ends of this straight line, there are deep emotional pits. If one lets their belief go too far to either end, they will fall into the deep pit. And when the evidence does accumulate against that concept or hypothesis, they will be unable to move towards the other end. We know some people who are already in the pit. Peter Duesberg is one. There are obviously others. I do hope that everyone involved in the XMRV saga, including Dr. Mikovits, will recalibrate and move away to a new position on the line before falling in the emotional pit. Keep up the fantastic work with TWiV. I will be listening in for years to come.
That's an excellent letter.
Yeah.
It's really, so I think this is important to add to my recommendations yesterday, that watch out for these emotional extremes as well. Really well put. So Vignac is actually a professor at Albert Einstein College of Medicine. He studies retroviruses. And the last one is from David, and this is for our panel. You guys will like this, and Janice as well. Mm-hmm. Guys is everybody. You know, in the US we call everybody guys, right? Mm-hmm. Dear TWiV, enjoy your podcast, but not being a virologist can have some disadvantages when it comes to trying to understand virology. It's a problem for us too, isn't it? Two questions for you that have been gnawing at me lately. One, I cannot understand how any truly novel virus can be discovered using tools that require knowing what to look for. PCR primers have to be made against a known sequence in order to amplify it. Antibodies have to be made that bind to antigens, which means you have to have the antigen you're looking for in hand before making antibodies to detect it. It's very much like generals fighting the last war. How can anyone claim to have found anything totally new and different if the tools and reagents are specific for what is already known? It's a which came first, chicken versus the egg issue. How can anybody find anything truly novel using tools depending on pre-knowledge? I can understand that these tools would be useful for finding similar viruses, but how do you use them to find something new? So someone here could probably answer that. Want to try?
I guess a good generic answer should be, when you're going to discover new viruses, I'm not a fisher, right? I just make a comparison. When you don't know, you throw a net, so you're fishing with a net. So you go for what is similar. When you want to go deeper, and we have found what's similar, you go to the difference. So first of all, you apply a generic method, and I'm not sure if I'm answering his email, but you try to be generic and unspecific, and then if you find something, you fish with a rod, right? You fish a specific difference.
Anyone else want to?
Well, I think these days it's actually we have lots of detection tools that do not require any prior knowledge.
Yeah, that's the net.
With the single-stranded DNA virus, the circular single-stranded DNA viruses, you have we call this rolled circle amplification. It uses an enzyme that will amplify any double-stranded circle in a plant. So it doesn't require any previous knowledge. And that's why we're finding new viruses all the time. And now you have metagenomics. You just sequencing everything. And then it doesn't require any previous knowledge anymore.
I mean, antibodies are more difficult because you have to sort of know what you're looking for. For some viruses, there are broadly reactive antibodies like retroviral envelope antibodies and so forth. But, and I guess for influenza, you could also make very pools of antibodies to detect everything, basically, right? All right, the second part of his question is, I just figured out how Western blots are supposed to work. But if they're prepared by running proteins down a denaturing gel, then transferred to a blot and finally probing with an antibody, doesn't the antibody have to be specific for the denatured protein of interest? Isn't it sort of like looking for an egg, the native protein, by detecting scrambled eggs? The protein after being denatured and transferred to a membrane for detection. So again, unless you previously know what an intact egg already looks like, how are you supposed to deduce anything about an egg in native form if the methods require you to scramble it up in order to detect them? I hope this question makes sense. Thanks for the podcast. Learning much. Signed, not a virologist.
However, this person is thinking like a scientist.
Absolutely.
You want to answer his question?
Well,
I don't know how
the
they work.
metaphor, the egg and the scrambled egg. I loved it.
It's a good one, yeah.
Looking for an egg, but looking for a scrambled egg.
I guess the answer is that antibodies that will work on Western blots detect denatured epitopes.
Linear epitopes.
Linear epitopes.
Not conformational.
Not conformational.
And you were just using the blot to detect the presence or absence of the protein. It doesn't say anything about how it is in the native state, right?
Still finding the scrambled egg.
That's right.
And assuming that comes from the egg.
Exactly, yes. Is that right? Everybody happy with that? Okay. All right, the last thing we do on TWiV is to do a couple of science picks of the week, and Grant and I will do that. I'll let you go first.
Okay, sure. My pick this week, I actually had a hard time making up my mind because there's several really great books I've read recently. But I'm going to pick The Disappearing Spoon. My wife just gave me a copy of it. It's by Sam Keen, and its subtitle is, and I love this, "And Other Tales of Madness, Love, and the History of the World from the Periodic Table of Elements." And it's really a series of tales about the various elements and people that have worked on it and how the periodic table evolved. And the title, The Disappearing Spoon, comes from a trick that many chemists use of making a spoon with the metal gallium. And gallium is a really interesting metal because at room temperature it's solid, but at 86 degrees Fahrenheit it melts. So one of the tricks that chemists do is to make a spoon out of gallium and put it in a cup of hot tea, and you can watch it dissolve. And if any of you are interested, if you just want to go to YouTube and type in melting gallium spoon, you can watch it. It's really cool. And the book is an awesome read. I highly recommended it. I've given my copy to Rich Condit, so if he gets around to reading it, you can ask him if he likes it, but any of you can pick it up. It's in paperback right now. Highly recommended.
You don't drink the tea afterwards.
No, in fact, gallium is a poison, so please don't drink that tea.
Be careful.
All right, great. I was wondering if your wife recommended it because you'd seen it.
That's for another TWiV, Vince.
My recommendation is a website at the American Academy for Microbiology. This is a division of the American Society for Microbiology. They have a colloquium program where they get experts together to study various important topics in microbiology, and then they write this up, and it's all free. So I'll put a link to the website in the show notes. They have PDFs of all of these colloquia, like clean water, bioinformatics and biodefense, everything to do with environment, food microbiology, genomics. It's really neat. These are great reports. They're a little technical, but they're free, and they're really neat in areas that you might be interested in. So check it out. It's the American Academy of Microbiology colloquia. We also have a listener pick of the week. This comes from Antonio, who says, "Hello there. activists. You get it? Activists. Thank you so much for these podcasts. I'm a Filipino programmer / tech geek who's been following the three of you since the Nile River virus episode. I've been absolutely captivated by this window you give into the world of biology and the life sciences. Your other listeners might find the Nature video covering the 2011 Nobel Laureates meeting in Lindau fascinating. There are 5 short films there featuring one-on-one dialogue between a student and a Nobel laureate who are both working in the same field, shedding insight on the passing of the torch between generations in science. And all the best from Antonio. So we'll put a link to that in the show notes. That's a video on these Nobel laureates at this very famous meeting in Lindau that they attend each year.
Another good one.
All right, so that will do it for TWiV in Brazil. We can be found on iTunes. We also have a website, twiv.tv. If you have an iPhone or an Android device, we have an app that you can use to stream the episodes to those devices. You can find it at microbeworld.org/app. And we love to receive your questions and comments. Send them to twiv@twiv.tv. I want to thank everyone for participating today, starting with Janice at the, well, I didn't get your institution right. Thank you for participating.
Okay, thank you.
Paulo at the University of São Paulo. Thank you. Thank you for participating. I got you in the wrong order. Don't take your mics off yet, though, because we have to finish a little business here. Francisco Zerbini at the Viçosa.
Like it's just an S, like an S.
You have to give me lessons afterwards.
I will.
Thank you for joining us.
Thank you for having me.
Eurico, thank you for joining us.
Thanks for having me.
And for letting us come here to Brazil.
Yeah, thank you for coming to Brazil.
Yeah,
you and Grant.
My pleasure. And Grant McFadden, University of Florida, thank you.
Vince, it's my pleasure, and I also just want to say on behalf of all virologists that what you and Rich and Alan and Dick do for virology and microbiology is fantastic, and I'd like to congratulate you.
Thank you. I'm, of course, Vincent Raccaniello. You can find me at virology.ws. I also want to thank the Sociedade Brasileira de Virologia.
Wow.
The organizing... I made so many errors, I'm sorry. The organizing committee, President Clarice Weiss-Arns and Eurico Arruda for having TWiV in Brazil. Thanks to the great audience as well. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.